Hello and welcome to our Molecular Networking for MetID tutorial. MetID, or Metabolite Identification Studies, are part of the preliminary safety testing for any new drug. The goal of these studies is to identify all the important metabolites of a new drug, especially any potentially toxic metabolites or metabolites which may affect the ADME or PK/PD parameters of the parent drug. FDA guidance for MetID studies focuses on phase 1 metabolism, which comprises a handful of biotransformations which are generally performed by the CYP family of human enzymes. However, this focus may miss important metabolites, especially those generated by microbial metabolism. The human gut microbiome encodes about 150 times more genes than are encoded in our own human genome. These include proteins which can regulate the activity of human drug metabolizing enzymes, as well as proteins which can directly act on and metabolize drugs themselves. This creates a whole new world of drug metabolites, many of which cannot necessarily be predicted before you begin your experiment. However, failing to identify these metabolites could lead to delays in drug development or even unexpected toxicity during clinical trials. So how do you identify drug metabolites if all of their potential biotransformations are unknown? This is where untargeted metabolomics paired with molecular networking can really shine. Untargeted metabolomics can measure all possible drug metabolites in a sample without having to predefine those metabolites in advance, while molecular networking can create a map of structure-related compounds so that you can rapidly identify your drug and all of its potential metabolites. So let's dive in and see how you can perform this type of study in Ometa Flow. So before you can identify drug metabolites, you first have to identify the drug itself. Well, this is generally done using library matching to MSMS spectral libraries. If you're looking at a novel compound, there's a good chance it's not already in your libraries. So we offer a workflow called MSMS Chooser that automatically finds library spectra in MSMS data. This is a fairly simple workflow, but if you want to learn more, you can always check out our workflow documentation. All you need to do is enter in a job description, select your input files, so these are your MSMS data that contains your library spectra, and then add an annotation file. Annotation files look like this, and this is just telling the workflow which file your library spectra is in, what is the name of your compound, what is the structure of your compound as a SMILES ID or its mass as M/Z, and whether the spectra was collected in positive or negative mode. This just tells the algorithm which compound adducts to search for. Once you've uploaded those two files, you simply need to choose your precursor tolerance in ppm or Dalton, and you can set a minimum precursor intensity, which will get rid of any really low abundance spectra. So this workflow is really fast. It completed in about 15 seconds, and once you're done, you can access all of your results here. For example, the annotations table gives you the list of every compound and compound adduct that was identified by this workflow. If you want to check out each spectrum, you can use this link here or visualize peak areas using our View LCMS browser. If any of these spectra are incorrect or you don't want to add them to your libraries for any reason, you can always use this delete button to remove that particular spectrum from your analysis. Once you've verified your results, you can add these spectra to your spectral libraries using this Add to Libraries button. This will pull up a new workflow where all you need to do is choose which library you want to add your spectra to. So once you've created those spectral libraries, you're ready for molecular networking. If you've never seen or interacted with a molecular network before, I highly suggest going over to our Running Your First Molecular Network tutorial, where we'll go into all of these options in a lot more detail. But for today, let's just check out how to use that custom library we've created in our molecular network. By default, molecular networking is going to use all your spectral libraries to perform library searching. But if you just want to use your custom library, you can click on the Select Spectral Library folder button here, find your custom library, and then click the Add Selected File to Spectral Library folder button here. You'll now notice that we have both all the libraries and your custom library selected, so make sure you remove the file selection for that LC folder. For today, the only other setting we're going to change is this Perform MS1 Peak Integration option here. We're going to change this option to Yes, so that we can run statistical analyses on our results. So this workflow took about 8 minutes to complete, and when it's done, you can access all of your results here. For this particular example, we're looking for microbial drug metabolites in about 70 samples that were created by incubating 8 common medications in a synthetic bacterial culture for 72 hours. Let's check out the molecular network using this button here. This will pull up your entire molecular network. In this network, each circle or node corresponds to an individual metabolite, and the edges between nodes indicates that there's structural similarity between those metabolites. If we scroll in, you'll notice that some of the nodes are blue and some nodes are gray. Blue nodes are metabolites with library matches, while gray nodes are unknown metabolites that didn't match to our library spectra. For instance, here we have a subnetwork which contains 2 matches for the drug ritonavir and then several unknown metabolites. To get an idea of where these metabolites are found, you can use the Show Pie Charts option here. This will color each node by which group it was found in. In this case, whether it was found in blank samples, samples that were collected at time 0, or samples that were collected after a 72-hour microbial incubation. Here we can see that ritonavir is mostly detected in the time 0 samples, but some of those structurally related compounds only show up after that microbial incubation. To get a better idea which metabolites were produced or degraded by these bacteria, we can run our statistics processing workflow. You can open up the statistics workflow by clicking on this button here, which opens up the workflow with all of the correct files. All you need to do is choose the metadata column that contains your groups of interest, whether or not to perform blank subtraction, and whether or not you want to run machine learning models. I want to get my results quickly today, so I'm not going to run those machine learning models. So this workflow took about 4 minutes to run, and now we can check out the results. The first visualization here is our volcano plot, which highlights metabolites that are up and down regulated at our different time points. To focus in on our potential drug metabolites, we can sort this table by the subnetwork that we know contains our drugs. Here we can see our matches to ritonavir and its potential metabolites. If we click on one of those metabolites, we can highlight that metabolite in the volcano plot and draw a box plot showing that this metabolite is upregulated after our 72-hour microbial incubation. There's a lot more to check out here, and you're welcome and encouraged to do so using the task links I've included in the video description. But for now, we've been able to create spectral libraries, perform library searching and molecular networking, and do statistical analysis in less than 15 minutes of total computational time. This type of screening can allow you to rapidly identify drug metabolites without having to determine all their potential biotransformations beforehand. Thanks for listening.